Very good morning to one and all. I am Dr. Nallagumar, working as a professor under Department of Pharmaceutical Analysis at SNS College of Pharmacy and Health Sciences, Coimbatore. I am handling for B.Pharm second sem students for POC-I, the Pharmaceutical Organic Chemistry 1 I am handling. In this Pharmaceutical Organic Chemistry, today we are going to learn about the isomerism. So it is not like traditional classroom activities. I am planning the design thinking based learning today. So this video is going to be very useful in this design thinking concept. Based on this design thinking concept, how we can learn the isomerism and types of isomerism and the major applications my students going to discuss. In this isomerism, we know this isomerism has same molecular formula and it has different structure. So, why we need to give such importance in this isomerism and what are the pharmacological activities will vary in this isomerism form we are going to learn. Before that, I am going to explain about the major classification of isomerism. In this isomerism, we have major two types. One is structural isomerism, another one is stereoisomerism. In this structural isomerisms, further classifications we can go for chain isomerism and positional isomerism and functional group isomerism and Metamerism, Tautomerism and ring chain isomerism form we have under this structural isomerism. So I already told the isomerism classification. It has major two categories. One is structural, the another one is stereo. In this structural isomerism, what are the further types? Okay, we classified already. So now I am moving to this stereotype isomerism. In this stereotype, okay, we have major two categories. One is geometric isomerism, another one is optical isomerism. So basically, in this pharma industry, we are consuming for citrusin. The molecule is very familiar, okay, we know. It has two isomerism form of the citrusin. One is levocitrusin, another one is dextrocitrusin. So if you are comparing these two different isomerism, whether it is dextrocitrusin or a levocitrusin, the levocitrusin has high pharmacological activities and it has very least side effect compared than dextrocitrusin. That is the only reason we picked that levocitrusin molecule for the treatment purpose. So my students going to explain how this isomerism playing major role in the drug discovery and what are the pharmacological benefits we can get from selecting this right isomerism format for the drugs or a API or a active content. And based on this isomerism, the molecule has different pharmacological activities and physiochemical activities. We need to understand the physiochemical activities is nothing but the boiling point of the particular drug or a melting point or a freezing point and other solubility. These are all playing vital role. So in the drug discovery and when during the drug discovery, we need to select for any solvent systems or we need to select for any synthesis module or we need to select suitable process for the drug synthesis. We need to know about these isomerism characters and along with the chemical properties also very important. So it is this isomerism has different physical properties and chemical properties as well as pharmacological activities. I quoted already this best example. The levocitrusin has best pharmacological activities compare than dextrocitrazine. And the quantity of drugs, okay, and loading dose, okay, we can reduce when we are selecting the better pharmacological isomerism form. This further discussion was done by my students. So I hope this TT session or the design thinking based learning is very useful for the viewers. Thank you.
Absolutely, sir. Isomerism matter because different isomers of a drug can have a different effect on the body. For example, one isomer might be therapeutically active while another could be inactive or even harmful. Like I read about thalidomide, one isomer helped with the morning sickness, but other gases have side effects, so pharmacists need to ensure right isomer is used.
I can say the problem is how can we design and synthesize drugs that constantly deliver the desired isomers to maximize therapeutic effect, benefits, and minimize risk. It's about precision in molecular design and ensuring the right isomer reaches the patient.
Chemistry view isomerism is critical in drug synthesis. If we don't control stereochemistry, we might produce a mixture of isomerism which can reduce a drug's efficiency or safety. In drugs like only one is active, so we need precise method is isolated or synthesis the correct isomerism.
Add that it also about cost and scalability. Producing a single isomers like through chain synthesis can be expensive and complex. So the challenges include finding a effective, co-effective way to get the right isomers.